Yeah, I'm very happy to introduce the next distinguished speaker, Piero Carninci, right? Dr. Piero Carninci is a PI at Riken, Japan. He's the Deputy Director of the Riken Center for Integrative Medical Science since 2018. Dr. Piero Carninci has developed technologies for transcriptome analysis, the CAP trapper, and the cage. And so far, Piero has published more than 500 papers in scientific journals, including Nature, Science, and so on. And Dr. Piero Carninci would give a talk, aging-related changes of circulating immune cells. Let's welcome Dr. Piero Carninci.
Right. Thank you very much, Gonhui, and also Aki for the kind introduction. Let's see if everything goes well. So you have been too kind in your introduction, indeed. And so what I would like to talk today is about some experiment that we started years ago, thanks to the collaboration of with Professor Hirose at Keio University. And Professor Hirose here, he has been collecting and creating a network of aging people, particularly supercentenarians. So supercentenarians are those people that can live 110 years or longer. And those are relatively few, no more than 200 people in the whole Japan. And only a small part of the centenarians become supercentenarians. So they are fairly healthy in their life and they age very healthy. So we are thinking with Kosuke, that has been in my lab for a long time, to analyze their blood, because it is very accessible, and see if we can understand anything about their secret for aging, looking at their immune cell, the immune cell from blood. So what we started to do, we started to collect blood. Here, the legend is SC means a supercentenarian, and CT are the controls, so 7 against 5, whole blood, peripheral blood mononuclear cells, and 10X. And in the first set of experiment, we just sequenced from the 3' ends just to look at gene expression. Then we went to Illumina, and then we started to analyze cells, as you can easily imagine. So, and good things is that we can identify cells, and actually quite a bit of cells, and we started to further analyze what are those. And by looking at specific markers, particularly CD3 and that are marked for T cells. So we identify this population. We identify many different populations, so we can easily assign, as you know very well. In particular, we focus on the T cells. So, and there is a cluster 1 and a cluster 2 that were grouped in a different way by Tisning. And we start to look at the difference between the supercentenarians and the controls. Supercentenarians here on the left, you see that this population, this cluster 2, is massively expanded if compared to the controls. Controls are adult people from in the age comprised from 50 to 60 and no more than 70. So there is a clear expansion of a group of cells. So we start to look a little bit more in detail. So first of all, we wanted to control if this is true for all the supercentenarian, and actually we see that the TC1 is low and the TC2 is high in all the supercentenarians. So this is quite common shared cellular phenotype. And then we wanted to look a little bit more at what happens in each specific cell. So again, we reanalyze with CIRA2. So cells here these cells are displayed in a different way, but our TC cluster 2 is here. And again, we can see that again, in the supercentenarian, this is highly overrepresented. than in the centenarian. We look at gene expression. And what was striking is that this cluster 2, there are those genes that has granzyme H, granzyme AB, granzyme A, and perforin. So those are cells, those are enzymes that are important for cytotoxicity. So cells having this enzyme are more cytotoxic, so they can attack cells and kill cells that they interact to. While in the other cluster that is more common in the non-aged people, those enzymes are not expressed. So, and again, we reanalyzed the data and we saw that those granzymes and perforin are broadly expressed essentially in all the subset of the cells. And this is not the case for the controls. And again, we look at how many of those enzymes are expressed in the supercentenarian, and clearly we see that most of the cells express at least 3 or 4 of those enzymes, so they are fully equipped as cytotoxic cells. Then we started to look at which class of T cells are important and are increasing this cytotoxicity property. And we look at the general population of the supercentenarian versus the centenarian. Clearly, the supercentenarian are more cytotoxic than the controls. But then we started to look at specific groups. And by analyzing other markers, so essentially we've been analyzing how many of these TC2 are CD8 or are CD4. And actually, we found that comparing CD8 and CD4, we found that the classes, the key class is the CD4, that usually in the controls, they're in the control donors, there are very few cytotoxic CD4, no more than few percent. They constitute 10, 15, 20, up to 50% of the cytotoxic cells, CD4 cells in the supercentenarian. So this is massively expansive, is massively expanded. It is very specific for the supercentenarian. We did some additional controls. We took additional samples. We looked at samples in their 20s, in their 80s, and we see that in the 80s there is a slight increase of granzyme beta in CD4 cells, and in the supercentenarian this is quite a prevalent class. So next we want to analyze, is this a broad population, or is this population of a few cells that are expanded? And actually we took advantage of the single cell VDJ kit. Essentially, we can prepare libraries and sequence those libraries from the 5' with the 10x is a kit of 10x genomics and analyzing single cell transcriptome. So we reanalyze and we look at we sequence the alpha and beta chain to see if those cells are clonal or are a very broad population. We call back 2 of the supercentenarians. The difficulty of this experiment is to call back the supercentenarian because actually we cannot call them. We need to visit them at their home and take a sample. And they are all over Japan, so we need to travel quite a bit with ice boxes to get those samples and then send back on time for the single cell analysis. And anyway, what was surprising here is that if compared to the helper cells, in the both of the supercentenaria, the number 1 and the number 2, we found that there are relatively few cells that are expanded and in many more cells. So in the first 69 in 908, in the second 385 in more than 2,200 cells. And relatively few of them, few of cells constitute between 75 or even 90% of the CD4 cytotoxic. So there are few that are massively expanded. And actually here is the numbers. So actually we found that how many each cell, each type is expanded in up to 500 or 700 cells in different donors. And we've been looking at all of them, because you will wonder, is there anything specific for all the supercentenaria? And this is from a different data set, including all this, including controls, centenarian and supercentenarians. And actually we found that all the chains are actually different. So there is not a single chain that dominates it, but actually there is quite a complex situation. We are still looking for what is the molecule that they bind. If there is anything shared, then probably we can discuss that later. So we say that they are cytotoxic. Are they really cytotoxic and functional? So we start to analyze their cytokines, and in particularly if they produce interferon gamma. This is from one of the supercentenaria, and actually those cells produce interferon gamma. This is supercentenarian 1 and 2, few cells, but still valid. So they also produce TNF alpha. So essentially, they seems to be totally equipped to be fully functional cytotoxic cells. So we started to, after publication of the paper last year, we started to further analyze, and we were wondering if we can understand a little bit better how those cytotoxic CD40 cells are expanded. Can we identify various antigens that they express in a time course? This is still a work in progress, and we don't have a final solution, final conclusion. But what we've been doing, we've been preparing a larger sample, single cell libraries, but also we added the detection of surface protein. So we had a series of antibody with a small barcode in order to identify what was the protein that is expressed on the surface of the cells, and also looking at the T cell receptor. We changed our sequencer, and it's a pleasure to also announce that the MGI sequencer has been working very well in our hands, and is also, we're very happy because it is fairly cheaper than the Illumina. Those are the number of the new set of samples. So we have new supercentenarians and new centenarians, but also a group of people between 70 and 90s, because we want to see the progression of those biomarkers from in the let's call normal aging into the supercentenarian aging. And we prepare libraries, and you know, it is all on the kit here, and those are numbered. But essentially, what I want to say is that we have been looking at various surface antigens and surface proteins that were analyzed by the antibodies and the barcode. Again, it's quite a lot of new data, transcriptome, T cell receptor, surface protein, all represented here, all sequenced with MGI. We sequence again and we isolated the CD3+, so essentially the T cells, extracted the CD3 plus the T cells, and we've been counting how many tags we have per sample. So essentially, all the sample yield at least 1,000 T cells, except one, but I think that we have quite a good number for the further analysis to see which kind of population of cells is represented, and count and make some of the statistics. By reclustering everything, and particularly reclustering CD3 plus T cells, 46,000 cells, so this is quite a large number, we confirm that the CD3 RNA is expressed everywhere. And also we take advantage also the all the remaining information from the experiment. And actually we divided in CD4 is the cytotoxic, and CD8 the helper, and actually they divide very well. We have additional classes of cells. Actually by selecting a large number, we can definitely improve the resolution. We look again to confirm at the granzyme beta and A and H, B and H here, and actually they are expressed in both classes. But in particular, we reanalyze and actually we look at this part here. It's essentially at the CD4 cells at this point. So far we've been talking about the CD4. Actually, here we will focus on CD8 in the coming slides. And actually, what we see, actually we see very little in the CD4 classes. So essentially by aging. So this color here is people in their 70s to 90s, centenarian and supercentenarian. The color code is maintained through all the slides. And actually what we see here is that there is not so striking evidence of changes of the population. Of course, we saw before the CD4 cytotoxic, the TAR increase. Then we look at the CD8, and actually what was evident was that the CD8 that are CD27 minus is continuously growing with age. So this is a second common features of the of aging centenarian into supercentenarian, and the CD27 is required for T-cell immunity. Is involving in many functions, including activator of NF-kappa B, MAPK8, JNK, and also in apoptosis. So actually, this is quite important, and we see that this population lacking CD27 is included. So this is a brief schema about what is CD27 doing, and again activating those important genes important for survival and proliferation of cells. I would like to briefly summarize, and I mentioned already all of this, and particularly we identify a class of CD4 cells that are cytotoxic, is highly enriched in supercentenarian, and CD27 is CD8 CD27 is expanded with CD27 minus expanded with age, and may mean loss of function on the immune system. Finally, I'm starting a lab, which is going to be my main activity over in the coming years at Human Technopole in Italy. If anyone may be interested in joining us, please contact me separately. I'd like to thank all the collaborators. Sorry for being a couple of minutes late. Thanking you all for all the attention, and happy to take any question. I'm not an immunologist, but I'll try to do my best. Thank you very much.
Mm-hmm. Thank you for the great talk. So from the result of your study, how do you think about why the centenarian can live so long? Have I any idea?
Only with the study is difficult to address the question, but well, for the specific aspect of the cytotoxic cell, we have been making many hypotheses. And one of the hypotheses is that a class of cytotoxic cells is specialized to prevent some common viral infections and probably or some common cancer antigen. And so we've been trying to set up some study to really identify the ligand, but it's not so easy to capture what would bind on the surface of those cells and isolate in enough quantity from human body and put it to mass spec. But we will really love if there's anyone interested in doing that that type of experiment. We will be very happy to further interact. The supercentenarians in general have they live healthy and in their 80s and 90s until they are 100 years old. Usually they don't develop any Alzheimer's disease, any dementia, but eventually when they reach 110 years old, they start to lose quite a lot of their intellectual ability, and eventually all of them started to show sign of dementia, and but but generally they live healthy. So, and probably there's something in their genes that we did not fully understand yet.
Okay, thank you. Could you please take a look at the chat? So to answer the question in that box.
Okay, Katie Borner, very interesting. How many supercentenarians were studied so far? What is the age distribution and men versus women? Yes, okay. How many supercentenarians? With this study, we have not expanded more than 12 or 13 or 14. Actually, the problem is that once you collect the supercentenarian, you cannot follow up them for years, because eventually very few of them go above 100, 115, rarely 117. So usually it's difficult to follow up, and age distribution, they're mostly women. Rarely there is a man, and most of them are women, and the age distribution is after 110. There are a few more, and then they drop down by 115, and so it seems we've been discussing what is the ultimate limit, and Professor Hirose believed that is no way to go above 120. Does it this answer? Okay, probably we are not.
We have a few questions actually, Piero. So if you could try to make them short.
Scott Walker, do you think a specific features of supercentenaria are hereditary, genetic, or are the result of their environment? There are probably quite a lot of the result of their environment, because the control very often is disposed. And actually, what Professor Hirose has been looking at is the children of the supercentenarians that now are in their 80s, and the children of the supercentenarian in their 80s are usually very, very healthy if compared to their spouses. And their spouses usually live in the same house, have the same lifestyle, same diet, they eat the same thing. So there is definitely some genetic component. Third question about inflammation level in supercentenarian. Inflammation level in supercentenarian and is usually low until the end. Usually, the increase of inflammation level is seen is seen just close, is dramatically increasing just closer to the end of their life, and in general they are less inflamed. Finally, at least in my list here, from is possible that expansion of CD4 cytotoxic is the result of aging, living so long, and not the reason for living so long? Yes, we've been discussing this very, very extensively. Is the CD4 really what makes us live longer, or them live longer? And well, it's difficult to say unless we find unless we find what do they bind. For instance, if they would bind some cancer antigen and prevent us to keep cancer free, we could probably make some better hypothesis. Otherwise, we don't know. But actually, those cells seems to be really functional. So this is the hope that they may have some kind of role. Right, I read in the question and answer. I'm not sure if there is more in the chat.
Yeah, actually, there is a couple of questions from Tommy in the chat.
Okay, I can go.
Oh, yeah. So the first question is, nice talk. Have you thought about looking at the epigenetic landscape difference between supercentenarian versus control, and with that associated to environmental factors such as food, living style, lifestyle, and so on?
Yeah, as we mentioned before, the children of the supercentenarians and their spouses usually show that it is not just diet and environment, because they live in the same environment. The children of the supercentenarian are more healthy on average than their spouse. Second, okay, I can read them. Submit questions to speak in a section. Okay, it's possible to perform metabolic and lipidomic analysis in these blood samples and associate this with your findings. Supercentenarian, actually, KU University has some blood collection, and probably we can cut on them to see that in this year, because of COVID-19, the sampling was stopped because given that COVID-19 is more dangerous for all other people, we decided, that Professor Rosa decided not to visit the supercentenarians in this year. So it's difficult to go back to the specific samples and sample that. Next from Hu Sun. Please,
no, maybe skip that. Yeah, last one.
What about the microbiome? We don't have associated microbiome for those samples. We don't have any analysis for microbiome associated for those.
But the Honda lab from the KO should have the microbiota data from the same patients, same supercentenarians.
Yeah, sure. Of course, we didn't analyze, but maybe for the discussion.
Yes, they have.
If you know anything, probably we can further discuss.
Yeah, that should be integrated. Yeah. Thank you, Piero.
Thank you, everyone. Thank you, Aki.
Thank you very much.